Look around the longevity and aging community. What were your signatures in immune project which told you it was aging?
In following individuals over the years, the most informative data type was proteins in blood.
The immune system tends to vary from the geographic locations, tends to vary a bit based on whether you're coming from a temperate country like India or a much more sterile Western Hemisphere country. To this, we call it the East-West or the North-South divide. In this situation, how do you see your immune age panel to vary between countries like India or somewhere? Do you think the signature baseline values will change? The AI wave is on. Everybody is doing AI these days, and we feel that AI is the only solution which we have for everything. How do you think this data collection, data curation, and annotation would lead to AI hallucinations in healthcare? How ready are we to use AI for healthcare? Hello, welcome to Biopic, where we talk about all things longevity, a podcast by Longevity India. And it's a pleasure to host today Dr. David Furman. Welcome, David. And it's a pleasure to have you here in India and talking to the Biopic podcast. So, quick introduction about David. David is a professor at the Buck Institute of Aging and is also Associate Director of Bioinformatics and Data Science Core. And it's wonderful to hear about a lot of things which David is doing in the 1000 Immunome project. And also, must mention David is also an entrepreneur and has got a couple of companies which is going to address things about aging and longevity. So let's start with some very quick questions. I mean, when we're looking about your history, you were born in Argentina and now you're in US and you had this interesting initiative called 1000 Immunomes. And tell us why you got interested in aging. We also saw somewhere that you started medicine and you quit medicine.
Yeah.
And then you've been doing research for decades now.
Yeah.
So please tell us your experience about getting into aging.
Absolutely. And thank you for inviting me. It's a really great pleasure to share with you this table and discuss aspects on longevity and aging. And I'm really pleased to see what you're building here in India. So when we started the 1000 Immunomes project, the idea was to understand the immune system in humans. For the most part, the knowledge about immunology has been based in animal models.
Yes.
And things that work in animals oftentimes don't translate to human biology. And so we decided to reinvent the wheel and say, let's study humans, right? And when we started analyzing a large number of data types, we were doing multiomics even before it was called multiomics, with proteins and cells and genes in blood from 1,000 individuals that we recruited as part of the 1000 Immunomes project. The realization was that there were aging signals all over the place. So we're looking at the immune system. We're looking at, in a large cohort of age range 20 to 100, and the aging signal was there, was constant, was giving us a lot of food for thought. And so that's where my interest in aging really started, from the idea of, okay, so we're looking at the immune system, but in essence, the signals are coming from aging. So let's start looking at the relationship between immune system, inflammation, and aging. And so that's how I got involved in the aging community. And later on, years later, actually 5 years ago, the CEO of the Buck Institute made me an irresistible offer to move up to Novato and lead the AI platform there.
Okay. So that's fascinating. So what you're saying is that the blood told you that it was aging and blood told you there is inflammation. And you just kind of connected them and while you were looking at immune.
Exactly. Yeah. By virtue of analyzing the data, we realized, that there's aging effect all over.
That leads me to a very fantastic question. Like when you look around the longevity and aging community, everybody keep talking about Horvath clock or this clock, and there's the immune age clock, there's a glycan age clock. What were your signatures in immune project which told you it was aging, not your favorite word exposome, which was triggering those inflammatory changes?
Um, so we analyze all sorts of different data types, proteomics from blood and cell frequencies and gene expressions, and we're trying to find determinants of health, right? And so in following individuals over the years, the most informative data type was proteins in blood. And we decided at that time to incorporate new tech that was coming up, deep neural networks applied to immunology, right? Never done before.
Yeah.
We learned from computer sciences. I became a data scientist by virtue of analyzing all these piles of data. I trained with Rob Tisciani, Deborah Hasty, Daphne Koller at Stanford, those who really invented machine learning and AI. And we were looking at all this data. Turns out that proteins were the most informative. When we analyzed the data in a nonlinear fashion with these deep neural networks, we could create a new clock, this inflammatory aging clock. It was capable of predicting multimorbidity, mortality, cardiovascular events, immune health. And so with Mark Davis, who I'm expecting will become Nobel Prize for discovery of T-cell receptors, we decided to form a company. It was too good to keep it in the academia, and we decided to bring it to the marketplace.
So that's interesting. So the immune system tends to vary from the geographic locations, tends to vary a bit based on whether you're coming from a temperate country like India or a much more sterile Western Hemisphere country. So this, we call it the East to West or the North-South divide. In this situation, how do you see your immune age panel to vary between countries like India or somewhere? Do you think the signature baseline values will change? Because we are curious because it's something very similar to what we would like to do.
Yeah, I think the expectation is that given that over 70% of the variance in the immune system is given by environment and not so much genetics, we have it was a Cell paper that we published in 2015 in diazygotic and monozygotic twins to decouple the effects of genetics and environment. Turns out that the vast majority, over 70%, was all environment. So if we're exposed to pollutants in the air or microplastics or water pollution or other sorts of exposures, I think the profiles will vary, right? Depending on those contaminants or pollutants and the individual responses to those. So your genetics plays a role in terms of the response to that. So I give you an example. The inflammatory age metric is a measure for your inflammatory health.
Right.
I could be 40 in my inflammatory age, but the way we get there is different. You could be also 40. The proteins that are determining that is different. For example, CXCL9 is a protein that is more involved in remodeling of the heart.
Right.
Naturally speaking, it is part of the immune system. But when it gets awry or constant and sustained, it will damage the heart. Now, eotaxin, a very different protein, will go more for the brain.
Right.
And the triggers are going to be also different. So when you are exposed to pollutants in the air, eotaxin will go up. So definitely, to your point, depending on the geographical location, and to some extent ethnicity, I would say we would expect different profiles.
In the baseline values.
The baseline values.
So that means there is a clear need to get baseline values from various parts of the world, define our
Absolutely,
data.
no doubt about it.
Great. So I'm so glad to hear that, because that brings what we are trying to do here at Longevity India makes it quite relevant in terms of understanding aging in a large mass of humanity which we have in India.
Completely, and in fact, as part of the 1,000 immunomes, we claim we have a measure for inflammation, but it really comes down to Palo Altonians, right?
Yes.
People from Palo Alto
That's very,
may not apply to other parts of the world. Yeah.
or even say for you, Argentina et al.
Yeah, exactly. Or African Americans or Asians or Indians.
Yeah. So the AI wave is on. Everybody is doing AI these days, and we feel that AI is the only solution which we have for everything. And do you think with a lot of this data which we collect in biological context, as you said, multiomics, you started doing multiomics before it became multiomics. And what we know that a lot of biological data is collected across different types of platform.
Correct.
That means there's an inherent noise on the system by which the data is collected. A lot of biological data is also collected by protocols which are not the best optimized protocol. How do you think this data collection, data curation, and annotation would lead to AI hallucinations in healthcare? How ready are we to use AI for healthcare?
So when we talk about hallucinations, we're referring mostly to large language models.
Yes.
Which is one application of AI. We have demonstrated that the more there is a human in the loop, the better and the less hallucinations we see. So I have a postdoc now who's a computer scientist who's working on deriving gene sets for different hallmarks of aging. And when she just run the algorithm without any supervision, we get 60% accuracy, which is really bad.
Yes.
Human in the loop goes to 85%. So there's definitely the need of having curation from human beings and training on curated data. That's one application, right? The way we're doing AI and machine learning applied to other topics is the multiomics.
Yes.
So you could not analyze the data without computers in the middle. I mean, if I were by myself, right, analyzing data, it could take me 100 years to analyze something that with a computer could do in a matter of hours, literally. And today we can read almost like 3.5 million papers during lunch. So
in terms of knowledge extraction.
Exactly. So it's a very different, very different endpoint and output that you can get from that.
So it's interesting what you say. So what AI brings is a platform to speed up the process of discovery. So how ready are we to use AI for addressing questions in aging and longevity? So because everybody wants to talk about longevity, about a magic pill, one pill at the end of the day, which resets our clock. Are we anyway headed in that direction? In our experience.
I mean, it's an interesting question because, I was chatting this morning with Matt, who is going to be part of the event.
Yeah.
And we both agree that the most successful treatments for extending health span and lifespan are 60 years old, right? Caloric restriction, for example, in mice, in animals, right?
Yes.
Now he overlays those results with rapamycin, right, or metformin, and we're not even closer to those early elements.
Okay.
So to what extent AI and machine learning has helped us intervention-wise, I don't think we're there yet. Diagnostics is very different.
True.
We now are measuring biological aging at large, and we can compute all that data. We have hundreds of thousands of measurements for each individual that goes into this pipeline, right? And the only way to interpret that is by using ML. So there's no way around that.
You mentioned about multiomics and you talked about diagnostics. So one question which probably would be asked to you on the panels day after tomorrow is the future diagnostics going to do the mass spec way? That you collect a whole bunch of data, without using individual testing platforms. And then we use AI to actually make sense of those set of data sets to predict what is going to be wrong for you in the future. That's something which your company Cosmica talks, what will happen to you in tomorrow. So is it the way diagnostics is going to go?
Um, I think it's not just AI/ML. It's also different technologies that are evolving at the moment. There's a really interesting now tool that uses quantum spectroscopy to measure the vibrational modes of every single molecule in a sample. So in essence, even isoforms of different proteins will vibrate differently. So for each sample, you can get around 150,000 features, metabolites, expression, proteins, the whole thing, including cell counts, for very, very cheap. So that's what keeps me up at night is, can we get to a point where we democratize biological aging assessment? And it's not just AI and machine learning, it's also other technologies that are evolving. What we do in Cosmica, and I'm going to allude to that in my talk, it's a little bit different. And the roots of it is that space flight accelerates aging.
Yeah.
So if you could model spaceflight in a lab, you can have cells spinning in this machine that was developed by NASA. And we buy these things from a Synthicon who has the license from NASA. And we accelerate the aging in 24 hours and have a window of how that person will age in the next 5 years. What are the mechanisms and what are the potential interventions that will work for that person?
That's interesting. So what you're saying is just change in microgravity, which is what Synthicon does, is sufficient to accelerate the normal trajectory. Now, what's interesting in this is we're missing the exposome.
Yeah.
So how do you account for that?
I love that. No, I love that question. If you don't do anything to your life, as far as lifestyle choices that have been discussed before, no, let's say you continue to do what you have done.
Okay.
That the assumption is you're not changing. We've done the experiment. We took longitudinal samples, baseline and 5 years later, of the same person who really aged, right? I take those peripheral blood mononuclear cells, so immune cells from blood. The original sample, you put it in microgravity, and compared to the older sample, it is 95% accurate at predicting that 5-year period. So what we're looking is the real aging can be accelerated in microgravity by about 5 years. If you use organoids, that's a completely different topic, and we're using that for drug discovery. Then, depending on the cell type, it will accelerate more or less. For example, brain organoids accelerate around 10 years biological aging. Cardiac cells, similar to blood, 5 years. And there's a ceiling.
This is all in the Synthicon setup.
Yes. And so the beauty of this is that I may age through inflammation, cell senescence, which are these two hallmarks of aging, but you may age in a different way, right? Cell-cell communication issues, telomere attrition, or whatnot.
Yes.
So that's what we can observe in 24 hours. We can have that window for that future aging and do something about it.
That's quite fascinating. So, but there's something which we always talk about, that you have the weak link in your body, the one organ which is at a different rate than rest.
Completely.
How do you handle that input in a microgravity experiment? Do we assume that the weak link information is already there in the system?
Not necessarily. Okay. So we see today, right? That's the baseline. And we see the incubation microgravity. That's the 5-year prediction. If we know that you're aging through a particular mechanism, we're going to propose using a gene compound enrichment analysis, which is like the whole interactome, which compounds will work to slow down your own aging process because you have this hallmark of aging that goes awry or is more affected than others. And that's using, again, AI and machine learning.
That's pretty interesting. So if I hear you right, David, what you're saying is that while there are n number of models to trigger aging in cells, which we know, right from radiation to chemotherapy drug, H2O to replicative exhaustion, what you're saying is that microgravity is another model and probably more accurate than what we know of other models, because other models have other complications associated with it.
Yeah, so that's a great question. So most of these models converge in particular pathways.
DNA damage response.
Exactly.
Yes.
So if you're hitting a cell with a radiation, you're going to be activating the DNA damage response, right? And very likely reactive oxygen species and mitochondrial dysfunction. It will vary across individuals because you're hitting one pathway.
Yes.
With the microgravity, you can see that it's the natural aging that's happening. We have those predictions. I'm going to show that the day after tomorrow in my talk. And so that doesn't converge in individuals. It's individual independent. So I may age through cell-cell communication issues. You may age through having more cell senescence.
You've started companies. Cosmica we just talked about. How about your other initiative, Edifice? Yes.
Yeah.
I mean, what's the premise of Edifice and Cosmica? How they're different? One is focused inflammation, and Cosmica is all about microgravity.
Yeah.
I mean, why don't have one?
So Edifice started from the work we did at the Stanford School of Medicine and the 1,000 immunomes. By analyzing all those data, we realized that there is a way that we can predict multimorbidity, mortality, and so on using 5 analytes. These are 5 proteins in the blood. So we combine everything into like 5 individual proteins. That is a metric for inflammation, which is one of the hallmarks.
Yes.
It's a relatively affordable test, so it has a different segment of the market. And it's pretty straightforward. As you know, you can order, and you have a Tasso device that you stick it in your arm. You get some blood drawn in these very small tubes, send that back to our labs, and that's done.
Okay.
Microgravity is quite different.
Yes.
Because one, it gives you a much precise view of how you will age. It may not be through inflammation.
Okay.
Right? It's much more expensive. It's a luxury product.
Yes.
Let's accept that.
Yeah.
It's a $2,000 per test, and that's the bare minimum, tier 1.
Yes.
We can also do compound screening in your age cells before you take any compound, right? So great example is my friend Brian Johnson, right? He takes over 100 different pills. And he's done a great job at lowering his biological aging, but maybe only 2 are working of those compounds.
Yes.
That's what we can unveil, right? With Edifice, it's not like that. It's more for inflammation.
Okay.
So different segments of the market, different applications.
Diagnostic therapeutics.
Exactly. More like we try to cover both aspects with Cosmica. With Edifice, is simply the diagnostic.
Okay. So that's interesting. So now coming back to what you talk about Edifice, you talk about inflammation. Inflammation has a protective role. In a country like India, I remember when I was doing my postdoc, and we will happily call it as chronically inflamed Indian. Because our inflammation is we just through the roof all the time. High IL-6, high IL-8, high CRP, low vitamin D, high cholesterol. It's surprising that we are still alive. But so do you think there is a difference baseline in inflammation for everybody, especially based on which part of the world you come from? It's again what I asked previously.
Yeah.
And how do you separate inflammation, which is protective, from inflammation, which is
I was gonna get to that. So we assembled a group of experts as part of a meeting that was held at Stanford in 2017, and the proceeding of that meeting were written in a chronic inflammation paper that was published in Nature Medicine 2 years later. What we ended up putting forward as a consensus is that there are no canonical markers of age-related inflammation. So when we look at acute inflammation, the response to the body to you cut your finger, it gets infected, gets red, swollen, warm. There are great markers in the clinic, sedimentation rate, you get IL-6, ERP, all those that you mentioned. When you look at age-related inflammation and you adjust for diseases, IL-6 is no longer there. CRP
may or may not be there.
Yeah, so today cardiologists will call CRP crap protein for the initials. Pardon my French, but it is not really a good marker for diseases of aging nor for aging itself. And so the whole purpose of the 1,000 immunomes was to derive new biomarkers of inflammaging. And that's what we did in the inflammatory age test.
So for a layperson, if I have to say that your inflammation is high and you say crap should be thrown away, what would be your next bet on? What would be your next set of panels, if you don't mind us sharing with us?
Yeah, so I'm biased, obviously, that I think inflammatory age is probably the only marker that has been demonstrated. SUPAR is another protein that has been demonstrated increasing with age. GDF15 is very constantly found. Not quite sure it's really inflammatory, but it is one of the proteins that is always increased in older adults with or without disease. So there's a variety of proteins, but it's not the canonical ones
Which would be looked
that are
to.
part of the acute. Yes, if you're looking at IL-6, you're looking at the wrong place.
It's nice to know that, because even in where we do research, this is the molecule which we go for.
I know.
Report IL-6, IL-8, and we're done.
Yeah.
So, and it's still an accepted way to look at age-associated inflammation.
Yeah, for less and less, I would say. Yeah.
So now, if you look at from chronic inflammation perspective versus a protective inflammation, which is in response to, let's say, COVID-19, which is where the things went completely south and everything just gone through the roof, dexamethasone seemed to work to control the inflammation.
Yeah.
Do you think something like that, a steroidal inflammation suppressant, which is like what rapamycin probably is doing, would be something which could take care of age-associated inflammation?
I think there are certain molecules that can hit inflammatory markers that are associated with aging. A variety of them, even statins. And rapamycin, other rapalogs that are targeting mTOR. And now urolithin A, which I think is coming up as a really strong.
Yes.
And then some other compounds like ketone bodies, ketone esters, are very strong at inhibiting inflammatory processes. In particular, a pathway that's called the inflammasome, right? And then after 3
Yes.
Vishwa Dixit at Yale has demonstrated quite a few of those papers.
Yes.
Demonstrating that there are certain things that can be targeted against those inflammatory pathways. Now, my take on this is it doesn't matter so much what you do in terms of fine-tuning that inflammation if you're not stopping the trigger.
Great.
Right?
Yes.
So the trigger being the
Exposome.
Exactly.
So that's a great segue to asking about the environmental contribution, the favorite exposome. Now, we live in a developed countries, and a lot of us live in developing countries and some underdeveloped countries. As you rightly said, exposome is very difficult to control because we live where we live. And so if you have to control exposome, what would be the easiest bet on? Like, would you say control the air, control the gut, control what you expose yourself into,
That's hard. I mean, I could spend hours about talking about the exposome, but in principle, any species that is moved to a new environment will develop inflammation.
Yeah.
And we're certainly not in our niche environment, in an ecological niche. So most things that we do to our bodies that are not aligned with the biology, with our genetics, right, will cause inflammation. Things from pollutants in the air, dysbiosis in the microbiome, all these surfactants that are in foods and in cosmetics, plastics and plasticizers, and then lack of being out there looking at nature, and then things like that are very obvious, but many times we don't talk about community, the sense of belonging. All these things, if we don't have them, you develop inflammation. Social isolation is one of the a very strong risk factor for increased inflammation, for high risk of heart attack in older adults and depression in young kids. So all these things accumulate, right, and are triggering different inflammatory processes. If we continue to live that way, there's no magic bullet that can solve that. So we need to stop the trigger, right? And that's another whole discussion, right?
Yes.
The elephant in the room, right? Because okay, so again, this is a conversation I've been having lately with many folks. It's like about longevity medicine. What can we do? Injection of peptides, exchange of plasma. That I'm going to show some of the results of a trial that we did, a therapeutic plasma exchange, and the many different modalities that longevity clinics do. Those are great, and some of them work, but you're being exposed all the background. You're breathing plastics. So I think one of the critical points is now, today, right this minute, you're filming, right, a podcast that will help people be more and more aware. This is exactly what we need. This is what will make the change, consumer behavior. And that's what's going to change the industry. That's what's going to change the policymakers. It doesn't start from policymaking. It doesn't start from regulation. It starts from consumers.
That's a very interesting point, David, but just got me curious from one aspect. The modern world has made everything small. In a sense, we just move around between one region of the world to another. And we take pride in doing that jet hopping, right? We just go around. And constantly there's a major flux of humanity from one part of the world to another to look for jobs, better opportunities, better environment. And coming from immune perspective, I said when we grow up, we get tolerated to the exposome because we get exposed to certain antigens or triggers when our immune systems are setting up. So do you think this changing environment is a big trigger for inflammatory phenotype later in the life? So you think if we have like the Okinawa there, you stay where you are, where you belong to, that could be one way to counteract age-associated complications?
Um, I would relate that more to early exposures of different sorts, infections, antigens, and so on, right? So the hygiene hypothesis. I think the microbiome is playing a substantial role in this. I would relate your question more as to if you get those microbiomes, right, early on that are protective, and you feed them with the soluble fiber and so on, and you have that diversity, that's largely protective, right? You decrease inflammation. It has been shown. We have a study where microbiome diversity is negatively correlated with inflammation, very clear. If you don't, and you're constantly cleaning your environment and getting rid of these bugs that oftentimes are protective, then you wipe out all your microbiome, and you're going to get inflamed, right? Other things that you mentioned, I would like to come back to the magic bullet idea that you mentioned, different anti-inflammatory compounds. For the most part, those work through inflammatory pathways that are in the acute phase, right?
Yeah.
And they hurt your microbiome. So I don't take, any of the ibuprofen or paracetamol,
Yes.
because I know it will damage my microbiome. It may resolve some inflammation at that minute, but in the long term, it will only increase it.
Yeah.
And so just wanted to kind of wrap that up into
So
the exposure.
because in a country like India, what we have here, we believe that everything starts at gut. So in Indian concept of healing says, fix your gut and you fix everything.
Yeah.
And we always talk about, using fibers and using fermented food to actually fix your gut before you actually start talking about everything. So we believe in gut brain, gut life.
Yeah, completely.
Everything comes with a gut axis built into it. So basically we are coming in our medical system a full circle now. So we started with micro breaking each organs and treating them. Now we're looking at a holistic, um, you know, uh, features of our body. Now, coming from that whole idea, so if you have to look at some signatures in somebody's gut to tell you about inflammation, because if we actually boil everything down to gut, what are the markers which you look for? Because again, gut biome changes from which part of the world you're coming from.
Completely. I love that question because we've been spending millions and millions of dollars in trying to identify the family of bacteria that are producing this and that, the combination of different microbiomes that can be producing that, and that's doomed. I think we're not investing the money from research, federal money, etc. I think it comes down to diversity, right? Um, we've seen that hunters and gatherers, for example, have 300 times the diversity than any other Western country,
Yeah.
right? Swedes, Italians, Americans, and so on. So I think it really comes down to having a great diversity of your microbiome, and you get that by exposing yourself. Don't peel your vegetables, right? Eat them as they are. Feed your microbiome with soluble fiber.
Yes.
So I think we're too much invested in understanding, identifying specific genus and families, and not so much looking
About
at
the
the whole
diversity.
picture. Yeah. So gut is super important. I agree with you. It's absolutely critical, but it's not everything, right?
Yeah.
There's also the skin, right? The microbiome of the skin, the microbiome of your oral cavity, that will largely determine inflammation in the oral cavity.
Yes.
And high risk for heart attack and Alzheimer's. It's been some studies linking that. We're just starting to realize how important diversity of microbiome in the gut, in the skin, in the oral cavity is.
It's what you say sounds music to me, because it, the traditional system of food habits in India also talk about diversity in food you consume rather than consuming large quantities of a single type of food. I think that's probably is true for most old cultures where we talk about fermented food and diverse type of food groups and largely plant based. So it's fantastic. So maybe one quick question: what is your go-to thing if you have to talk about your longevity, or what is the thing you would like to do?
Well, I mean, it's a long protocol, but I try to. I'm not saying I won't never do this, right? Because it's very difficult to stay away from pollutants and plastics and things like that. But say, for example, um, my toothbrush is boar bristles
No, okay.
in a wooden stick,
What is take?
right? I don't put those plastic
Plastic
toothbrushes. we stop using plastics as containers, right?
Okay.
We don't do any of the dairy products. I know here in India it's very, very common.
Yes.
But we didn't really evolve consuming dairy products. We domesticated cows only 4,000 years ago. Same for wheat, right? I don't touch any wheat. And so it's a long list. And then it comes down to, like, for example, hygiene. How much do you go to the shower and use shampoo? All those products are killing your microbiome. And so, I like to just use water, right? The water filters that you have are also plastics. So instead of using water filters, I boil the water and you have sediments. And you take that as
It's almost like a therapeutic regime of
think of these hunters and gatherers.
Yes.
Right. The more you can resemble that type of life, provided that you're in a very difficult, very different world, I think the better you're going to be. My inflammatory age went down significantly. There's a Wall Street Journal note, an article, an interview that will come out very soon on a anti-inflammatory protocol that I did for 4 years. It was very, very strict. Eating basically wild-caught salmon and organic berries. My inflammatory age went down by 10 years. And so it was very significant.
almost like living in the wild.
It was completely off grid, off the grid. But it didn't, it's a complicated life. So then had kids, and then they had to go to school. So this is a beautiful place that is 30 minutes towards the beach from the Stanford campus. It's called San Gregorio Valley.
Okay.
And they had this beautiful cabin there. And I became basically almost like a hunter and gatherer.
Yes.
And it was a really great experience.
It's fascinating you mentioned that, because in ancient India we say the people who live longer, the one who lives in caves, will live like hunter and gatherers, and also breathe much lesser just by practicing controlled breathing.
Yeah, well, there's good data showing controlled breathing lowers inflammation.
So it's fascinating what you mentioned, because there's something which I argue with a lot of people all the time. That the design of nature is what you can consume in large amounts is easily accessible, like fruits. What nature did not want you to consume in good amount is actually makes it difficult. Like, look at the wheat. You got to spend a lot of effort to take the thing out. So you should not be eating that much.
Fully agree.
So that's pretty interesting that,
follow the evolutionary roots.
Yes. So let's come to what you have on your wrist too. How much you rely on the wearables to track, and what are you looking for when you use a wearable device in terms of understanding your health or your trajectories of health?
Yeah. So it's very useful because it gives me this biological feedback, right? I know pretty much what's good for me and what's not so good for me. I tracked my glucose with a continuous glucose monitoring device. I'm not using it anymore because I know what spikes my glucose. So now I learned, right? But I wear the Whoop device because when I exercise, I want to have a minimum of 10 of strain, which is one of the metrics that Whoop gives you. If you don't have that, you don't know where you're at. And so for me, the bare minimum daily strain has to be over 10. And the only way to measure this is by, using a wearable. Yeah. If you don't measure, you can't change it.
That's interesting, because that's that narrative I heard many places. You got to measure to fix it. If you don't measure, that you do not know what you're fixing.
Completely.
So now, if you have to spin this narrative for a country like India, where a large majority of people are, we do not on a richer category, they're actually middle or lower middle. They'll come, and 70% live in the rural settings. If given a choice, a mass deployment device or something, what would you bet and what should they measure? Or what should we measure as a country to give an index or measure of health? Since you have played around from molecules to systems, it is something worth hearing from you.
It's a bit of a complicated question. I don't think there is currently in the market a system that can accurately measure your biological aging rate as in an implantable device that can give you, continuous monitor of that. And there's if they exist, they're fairly expensive,
Yeah.
right? And so how do you capture this? Okay.
Or it's just anything.
Yeah. So, I'm doing in my research, I have several groups, right?
Yes.
From the group that is doing the most sophisticated prediction of aging to those that are trying to democratize it.
Yes.
I want to talk about the healthy selfie for a moment. So we collected organ aging metrics in 750 people in a study that we run internally in my company. That's Edifice Health. We paired that with facial images.
Okay.
So now I'm applying AI to predict organ aging based on the facial image.
That's interesting.
So, and we have really good data for IH. Now we're going to see if we can predict other organs or other parts of the body aging, right? And so if we're successful at doing that, that means that it becomes basically almost free, right? If you can make it free for everybody, just having a cell phone could give you an idea on where you're at in your aging rates.
Yes.
And you can do things to modify that. This is an ultra low cost assessment of diagnostics of aging. Right?
Brilliant. Brilliant. This is the perfect solution for a large, complicated question I asked. So last three quick fire questions for you. There's so many biological clocks around us.
Yeah.
Harvard, this and that. If there's so many clocks, my gut feeling says all of them are wrong. Because if there's so many clocks and they're all showing different time, none of them are right. What's your bet?
Okay, so there are first generation clocks that are trained or learning their models based on chronological age. I think those are outdated.
Yeah.
There are second generation clocks that are trained. It doesn't matter what you're using, if epigenetics or metabolomics, or
Yes.
but are trained in mortality. These are essentially mortality predictors. Those are much more useful. And there are those who are built based on trajectories at the individual level. So not so much cross-sectional with a population, but following people over time and fitting models to learn, for example, what's called pace of aging, the Dunedin study.
Yes.
Like that one to me is one of the most accurate ones. In my hands, is the most sensitive ones. Inflammatory age is focusing on the inflammation, but when you talk about more broadly biological aging clocks, I really like the pace of aging because it's been trained on trajectories and not so much cross-sectional data. So I think my take is that's probably the most accurate one.
So if you do not have the trajectory, I'll go with grim age.
Grim age is also very good. I agree. Pheno age is also pretty decent, but I'm going to present disease age. Which is now under review in Nature Medicine. We're going to expect to publish soon. So we have it a preprint at the moment, and I'm going to present that in my talk the day after tomorrow.
Next quick question. Supplements. Which are the hyped one, which are have some value? We talk about urolithin A.
Urolithin A, I think it's coming up. It's very promising. Senolytics are actually pretty decent in terms of reducing a number of different biological aging metrics. So quercetin and ficitin are powerful.
DQ?
Yeah, exactly. I think it's also pretty decent, like dasatinib and quercetin. So all those family of senolytics and xenomorphics. And now the one that I really like as well, that hasn't not been demonstrated so much, but we have now clinical trials at the Buck Institute, is ketone esters.
Okay.
So that's very promising. So wait to hear results from that. Hype. I think resveratrol is one of Larry's favorite. Yeah, exactly. I think it's failing to us. What others? Acid derivative elements.
Yes. Yeah.
And then yeah, those probably are the ones that
Just hybrid one.
less interesting.
That's fantastic. One last question.
Sure.
Where do you think the aging industry is going to change in the next 5 to 10 years? Where are we headed? What's going to change? Is it going to be test and fix? Medicine 3.0, 4.0? We talk about that also.
Let's see. I think there's going to be a transition from a luxury products and services for the very wealthy, worried well, we call them, to more products and services that can be implemented in the general population. At least that's my hope. That's what I am pushing for. So we're now building a project on longevity cities. Okay, right, with Nick Palmieri and Lynn Corner at the UK, in the UK. It also comes down to your household, to designing your workplace, exactly, to address the exposome, right? So the many fields now are converging. Architecture, which who would have thought? Exactly. Now it's getting more and more developed, and so I think there's going to be a transition towards more of a democratizing the diagnostics and interventions for aging. They're going to become cheaper. The moment we can bring this to the FDA, and there's a path for that, it's going to become basically reimbursable, right?
Yes,
and free for everybody.
Exactly.
So the WHO has recognized a decline in intrinsic capacity associated with aging as a disease in the ICD-11, International Classification of Diseases 11, under the code MG2A. So that means that this opens the dialogue with the FDA to develop one, diagnostics, two, interventions for a decline in function. And so if we can bring these to the FDA, get FDA approval, and get a CMS code, we can get reimbursed. And then everybody else will follow.
Yes. Thank you so much, David. Thank you.
Absolutely.
I mean, it was wonderful talking so many different things about aging with you. We look forward to hearing the more technical talk in our longevity summit. And we really hope that we can keep talking about more interesting things in
a lot of areas of collaboration I can see. And thank you so much for having me.
Thank you so much. Wonderful. So that brings end to this session of Biopic Live. Please keep looking forward for other podcasts and other talks with very interesting people who talk about all things in longevity and aging.